Lipid biosynthesis. In this 3-lecture series, we discuss how lipids are made. You should have briefly touched on this topic in Biochemistry 1. In Biochemistry 1, you learned that lipid catabolism occurs in the mitochondria of eukaryotic cells, where the citric acid cycle is present to oxidize the generated Acetyl-CoA for oxidative phosphorylation. You should have also learned that lipid synthesis occurs in the cytoplasm. Thus, the two pathways are spatially isolated from one another, so that there is no direct competition between the generated and consumed metabolites. The learning goals are biosynthesis of fatty acids, assembly of fatty acids and glycerol into triacylglycerols, biosynthesis of cholesterol, and mechanism of fatty acid synthase. Lipids fulfill a variety of biological functions. These include energy storage in your adipocytes, constituents of membranes, anchors for proteins, myristoylation, cofactors for enzymes and protein folding, signaling molecules such as in PIP and PIP2, pigments, the colors of birds, detergents, transporters, you should remember emulsification, and antioxidants, for example, vitamin E. Catabolism and anabolism of fatty acids. These processes are performed by different pathways and in different locations of eukaryotic cells. Catabolism of fatty acids generates Acetyl-CoA, NADH, and FADH2. This occurs in the mitochondria. Anabolism of fatty acids requires Acetyl-CoA, the reducing power of NADPH, and takes place in the cytosol of animal cells and chloroplasts of plant cells. The chemistry of lipid synthesis is very similar to catabolism of fatty acids, but there are important differences. Subcellular localization and lipid metabolism. Yeast and vertebrate cells differ from higher plant cells in the compartmentation of lipid metabolism. Fatty acid synthesis takes place in the compartment in which NADPH is available for reducing reductive synthesis, i.e., where the NADPH over NADP plus ratio is high. This is the cytosol in animals and yeast cells, and the chloroplasts in plant cells. Processes in red type are covered in this chapter. Overview of fatty acid synthesis. Fatty acids are built over several iterations of the same chemistry using malonyl-CoA, which is a modified acetyl-CoA. Each iteration involves reduction of a carbonyl carbon to a methylene carbon. We will discuss how acetyl-CoA is converted into malonyl-CoA. Fatty acid synthesis occurs in cell compartments where NADPH levels are high. So where does NADPH come from? A couple of slides ago, I mentioned that NADPH is used for synthesizing lipids. In Biochemistry 1, we learned that the pentose phosphate pathway was one of the pathways to generate NADPH. Plants can also create NADPH using photosynthesis. Pathways for NADPH production. Oxidation of malate by malic enzyme using NADP plus generates pyruvate, NADPH, and releases carbon dioxide. Glucose 6-phosphate is converted to ribulose 5-phosphate in the pentose phosphate pathway for generating 2 NADPH molecules. The acetyl-CoA carboxylase reaction. Acetyl-CoA carboxylase has 3 functional subunits, or chains: biotin carrier protein, biotin carboxylase, and trans-carboxylase. The image on the right is an outline showing the quaternary architecture of acetyl-CoA carboxylase. The biotin carrier protein is in the center. Attached to this protein via a lysine side chain is a biotin that is carboxylated. This process is carried out by biotin carboxylate, colored as blue on the left-hand side of the image. Biotin carboxylase requires hydrolysis of ATP to ADP plus inorganic phosphate and uses bicarbonate from the cellular milieu. This process activates the CO2 for chemical modification. The long, flexible biotin arm of the biotin carrier protein carries the activated CO2 from the biotin carboxylase region or domain or subunit to the trans-carboxylase active site, colored in black, on the right-hand side of the image. Trans-carboxylase transfers the activated CO2 from the biotin to Acetyl-CoA to generate Malonyl-CoA for lipid biosynthesis. Biotin carboxylase activates CO2 by attaching it to the nitrogen of the biotin ring in an ATP-dependent reaction. Trans-carboxylase transfers activated CO2 from biotin to Acetyl-CoA, producing Malonyl-CoA. The long, flexible biotin arm carries the activated CO2 from the biotin carboxylase region or domain or subunit to the trans-carboxylase active site. There are two steps in this reaction. The steps are similar to carboxylations catalyzed by pyruvate carboxylase and propanoyl-CoA carboxylase. You've seen pyruvate carboxylase and propanoyl-CoA carboxylase involved in feeding metabolites into the citric acid cycle. In part 1 of this reaction, CO2 is activated by attachment to the nitrogen in the biotin ring. This requires ATP hydrolysis and produces carbamol. In the second part of the reaction, biotin carrier protein undergoes a conformational change to carry carbamol to the trans-carboxylase active site. This process is called substrate channeling, and you probably first saw it. The synthesis of fatty acids is catalyzed by fatty acid synthase, or FAS for short. This reaction is a 4-step sequence that involves the addition of 2 carbons to a growing fatty acyl chain. Each malonyl group and acetyl or longer acyl group is activated by a thioester that links it to fatty acid synthase, a multi-enzyme system that will be described later. Reaction 1 is the condensation of an activated acyl group and 2 carbons derived from malonyl-CoA, with elimination of CO2 from the malonyl group. This extends the acyl chain by 2 carbons. The beta-keto product of this condensation is then reduced in 3 more steps that are nearly identical to the reactions of beta oxidation, but run in the reverse sequence. In reaction 2, the beta-keto group is reduced to an alcohol. In reaction 3, there is an elimination of H2O to create a double bond. And in reaction 4, the double bond is reduced to form the corresponding saturated fatty acyl group. The term FAS I is used for FAS of vertebrates and fungi, and FAS II is used for FAS of plants and bacteria. FAS I versus FAS II. FAS I is a single polypeptide chain and produces 16 carbon-saturated fatty acids. The C15 and C16 are from the Acetyl-CoA used to prime the reaction. This is the initial step of the reaction. Remember that growth occurs at the head, not tail of the fatty acid. FAS II is made of separate diffusible enzymes. This makes saturated, unsaturated, branched, and many-legged fatty acids. Fatty acid synthase type 1 systems. Shown on the left-hand side is the mammalian FAS I, a homodimer. Shown on the right-hand side is the fungal FAS I, a heterododecamer. Each chain of the mammalian FAS I is composed of multiple domains, shown as colored boxes or ellipses in the scheme below. The names of these domains also indicate their enzymatic activities. These are beta-ketoacyl ACP synthase, abbreviated as KS, malonyl acetyl-CoA ACP transferase, abbreviated as MAT, beta-hydroxyacyl ACP dehydratase, abbreviated as DH, enol ACP reductase, abbreviated as ER, and beta-ketoacyl ACP reductase, abbreviated as KR. ACP is the acyl carrier protein. TE, the seventh domain, is a thioesterase that releases the palmitate product from ACP when the synthesis is completed. The ACP and TE domains are disordered in the crystal and are therefore not shown in the image. In the structure on the right is that of a fungus FAS I system. The same active sites are divided between two multifunctional polypeptide chains. 6 copies of each polypeptide are found in the heterododecameric complex. A wheel of 6 alpha subunits, which include ACP, as well as the KS and KR active sites, is found at the center of the complex. In the wheel, 3 subunits are found on one face and 3 on the other. On the other side of the wheel are domes formed by trimers of the beta subunits, containing the ER and DH active sites, as well as 2 domains with active sites analogous to MAT in the mammalian enzyme. The domains of one of each type of subunit are colored according to the active site colors of the mammalian FAS I. Fatty acid synthesis. The overall goal is to attach an acetate unit, which is 2 carbons, from malonyl-CoA to a growing chain of carbons. Then reduce this product. Again, this is similar to the chemistry you've seen in fatty acid oxidation, but run in the reverse order. The reaction involves cycles of 4 enzyme catalyzed steps: condensation of the growing chain with an activated acetate, reduction of carbonyl to hydroxyl, dehydration of alcohol to transalkene, and reduction of alkene to alkane. The growing chain is initially attached to the enzyme via a thioester linkage. Step 1 of FAS I, elongation. The figure concentrates on the chemistry of fatty acid synthesis. FAS I is shown as a stack of disks. We will go over these steps multiple times, each time with more information included. We begin by having FAS I loaded with its initial reactants, the acetyl and the malonyl groups. We will cover these steps in later slides. The first reaction is the condensation of the acetyl group with the malonyl-CoA to eliminate a CO2 from the malonyl group. This extends the acyl chain by 2 carbons by transferring the acetyl group to the decarboxylated malonyl group. The mechanism of the first step of this reaction is given to illustrate the role of decarboxylation in facilitating condensation. The SH or thiol that was attached to acetyl group is now free to bind to another acetyl group. The beta keto product of this condensation is then reduced in 3 more steps, nearly identical to the reactions of beta oxidation, but in the reverse sequence. In step 2, the beta keto group is reduced to an alcohol using the reduction power of NADPH. In step 3, elimination of H2O creates a double bond. In step 4, the double bond is reduced using the reduction power of NADPH to form the corresponding saturated fatty acyl group. The overall process of palmitate synthesis. The fatty acyl chain grows by 2 carbon units donated by activated malonate, with loss of CO2 at each step. The initial acetyl group is shaded yellow. The acetyl carbons correspond to carbons 15 and 16 of palmitate. The C1 and C2 of the first malonate are shaded light red. The malonate carbons correspond to carbons 13 and 14 of palmitate. The carbon released as CO2 is shaded green. This is released at each step where addition of an acetyl group is performed. After each 2-carbon addition, reductions convert the growing chain to a fat-saturated fatty acid of 4, then 6, then 8 carbons, and so on. The final product is the 16-carbon fatty acid palmitate. Acyl carrier protein, or ACP for short, serves as a shuttle in fatty acid synthesis. Again, the concept of substrate channeling should not be new to you. Acyl carrier protein serves as a shuttle in fatty acid synthesis. ACP is a domain of FAS I. ACP contains the covalently attached prosthetic group 4-phosphopentanethane to the side chain hydroxyl of a serine. This flexible arm is modified at the terminating thiol and is responsible for delivering reactants or products from one enzyme active site to another. It delivers acetate in the first step of fatty acid synthesis and malonate in the remaining steps of fatty acid synthesis. It also shuttles the growing chain from one active site to another during the 4-step reaction. You will see these steps in slides to come. Charging ACP and FAS I with acyl groups activates them. 2 thiols must be charged with the correct acyl groups before the condensation reaction can begin. These are the thiol from 4-phosphopentanethane in ACP and a thiol from cysteine in fatty acid synthase. The acyl group of Acetyl-CoA is transferred to ACP. This reaction is catalyzed by malonyl or acetyl-CoA transferase, or MAT for short. ACP passes this acetate to the cysteine of the beta-ketoacyl ACP synthase, or KS for short, domain of FAS I. ACP-SH group is recharged with malonyl from malonyl-CoA. Charging, activation with ACP, and the 4 steps sequence of mammalian fatty acid synthesis. FAS I is simplified to a little snake with its multiple domains labeled and colored. The domain names will be reduced to the shown acronyms for simplicity. For example, beta-ketoacyl ACP synthase will be referred to as KS, and malonyl acetyl-CoA ACP transferase will be referred to as MAT. We begin with the FAS I uncharged. Note that we have 2 free thiols, and both of these will be charged as FAS I synthesizes fatty acids. For each step, only the domains completing the indicated function will be colored. In step 1, MAT will charge the thiol of KS with an acetate. The acetate comes from acetyl-CoA. This reaction releases CoA, which is used in other reactions. In step 2, MAT will charge the thiol of ACP with malonyl, again releasing CoA into the cytoplasm of the cell. Note how ACP is able to swing from one domain to another for substrate channeling. FAS I is now charged with an acetate on the thiol of KS and the malonyl on the thiol of ACP. In the next step, a condensation occurs between the malonyl attached to ACP and the acetyl attached to KS. The acetyl group from KS is transferred to the terminus of the red carbons in malonyl. This releases a CO2 from malonyl to generate the beta-keto-butyryl ACP shown at the bottom of the slide. The CO2 is a relatively good leaving group and plays an important role in driving the reaction. The ACP now swings from the vicinity of KS to the vicinity of Kr. Beta-keto-butyryl is a molecule you've seen in Biochemistry 1 when you were studying fatty acid oxidation. Kr uses the reductive power of NADPH plus H plus to reduce the beta-ketobutyryl ACP to beta-hydroxybutyryl ACP. Again, this is the opposite of what you saw in Biochemistry 1, where fatty acids were oxidized to generate Acetyl-CoA. The NADPH plus H plus is oxidized to NADP plus. In the next step, ACP swings from Kr to DH, and the beta-hydroxybutyryl ACP is dehydrated to trans-delta 2 butanoyl ACP. Again, this is the opposite of a step in beta oxidation of fatty acids. This process releases a water molecule and generates a double bond between the carbons that were part of the Acetyl-CoA and malonyl-CoA condensation at the very beginning of fatty acid synthesis. ACP then swings to ER for the next step. ER uses the reduction power of NADPH to reduce the double bond to generate butyryl ACP and NADP+. At this point, you have used two NADPH molecules. ACP now swings back to Ks. ACP is again near Ks as it was near the beginning of the cycle. However, Ks now has a free thiol that needs to be charged in order for fatty acid synthesis to continue. The butyryl group on ACP is transferred to the free thiol of Ks. ACP now has a free thiol. As of this point, ACP has interacted with every domain on FAS I. You've seen something very similar to this already and should know what is to follow next. MAT grabs a malonyl-CoA and attaches the malonyl to the thiol of ACP. Again, this process liberates a CoA which can be used in other reactions. Note that while the figure on the bottom shows an acetyl group attached to Ks, this step involves a butyryl group attached to Ks. In summary, the elongation step involves a condensation with acetate by beta-keto-acyl ACP synthase, or Ks. Then a reduction of carbonyl to hydroxyl is performed by beta-keto-acyl ACP reductase, or Kr for short. Following this, a dehydration step by the beta-hydroxyacyl ACP dehydratase, or DH for short, generates an alkene. The alkene is reduced to an alkane by enoyl ACP reductase, or ER for short. The elongated fatty acid chain is then transferred from ACP to Ks by malonyl acetyl-CoA ACP transferase, or MAT for short. Stoichiometry of synthesis of palmitate. The reactants are 7 malonyl-CoA, 1 acetyl-CoA, and NADPH. Each malonyl-CoA is synthesized from an acetyl-CoA and bicarbonate using the power of ATP hydrolysis. 7 cycles of condensation, reduction, dehydration, and reduction correspond to a total of 1 acetyl-CoA, 7 malonyl-CoA, 14 NADPH plus 14 protons generate 1 palmitate, 7 CO2, 8 CoA, 14 NADP, and 6 water molecules. In Biochemistry 1, you learned that Acetyl-CoA is made in the mitochondria of non-photosynthetic eukaryotes. Given that fatty acid synthesis occurs in the cytoplasm of these cells, the Acetyl-CoA needs to be shuttled from the mitochondria to the cytoplasm. This process requires 2 ATP molecules per acetyl-CoA. Therefore, the cost of fatty acid synthesis is 3 ATP molecules per every 2 carbons added to the growing fatty acid chain. What molecule is responsible for generating the acetyl-CoA molecules in the mitochondria that ultimately end up in the fatty acids synthesized in the cytoplasm? Shuttle for transfer of acetyl groups from mitochondria to cytosol. The outer mitochondrial membrane is freely permeable to all these compounds. Pyruvate derived from amino acid catabolism in the mitochondrial matrix or from glucose by glycolysis in the cytosol is converted to acetyl-CoA in the matrix. Acetyl groups pass out of the mitochondrion as citrate, and in the cytosol they are delivered as acetyl-CoA for fatty acid synthesis. Oxaloacetate is reduced to malate, which can return to the mitochondrial matrix and is converted to oxaloacetate. The major fate for cytosolic malate is oxidation by malic enzyme to generate cytosolic NADPH. The pyruvate produced returns to the mitochondrial matrix. We begin with pyruvate, either in the cytoplasm, bottom right, or mitochondria, bottom left. The pyruvate in cytosol is transported into the mitochondria via the pyruvate transporter. In the mitochondria, pyruvate carboxylase attaches the CO2 to pyruvate using ATP binding and hydrolysis to create oxaloacetate and ADP plus inorganic phosphate. Citrate synthase, you've seen this before, picks up the acetyl-CoA generated in the mitochondria and modifies oxaloacetate to generate citrate and CoA. Citrate is transported into the cytosol using a citrate transporter. In the cytosol, citrate lyase uses CoA to modify citrate to generate acetyl-CoA and oxaloacetate. This step uses ATP hydrolysis. The oxaloacetate is reduced to malate using the reducing power of NADH plus H plus by the enzyme malate dehydrogenase. The malate is oxidized as decarboxylated by malic enzyme to generate pyruvate. And this is done using the oxidizing power of NADP plus. Some of this malate is also transported into the mitochondrial matrix via the malate alpha-ketoglutarate transporter. Now remember that the CoA in the matrix is distinct from the CoA in the cytoplasm. Fatty acid synthesis is tightly regulated. You must have seen something like this in Biochemistry 1 when you were studying fatty acid oxidation. In the cells of vertebrates, both allosteric regulation and hormone-dependent covalent modification influence the flow of precursors into malonyl-CoA. Acetyl-CoA carboxylase, or ACC, catalyzes the rate-limiting step for fatty acid synthesis. This is the conversion of acetyl-CoA to malonyl-CoA. Glucagon and epinephrine trigger phosphorylation of ACC. This slows down the activity of ACC and produces less malonyl-CoA for fatty acid synthesis. High palmitoyl-CoA concentration downregulates ACC via a feedback inhibition mechanism. In contrast, high concentrations of citrate and acetyl-CoA indicate that the energetics of the cell are being met and that fatty acids or energy storage should be turned on. Regulation of fatty acid synthesis in vertebrates. Phosphorylation of ACC turns it off. ACC is inactive as phosphorylated monomers. When dephosphorylated, ACC polymerizes into long, active filaments. Phosphorylation reverses the polymerization. Fat, triacylglycerols, and phospholipids serve multiple functions in animals, plants, and bacteria. Phospholipids and triacylglycerol possess a glycerol backbone and 2 to 3 fatty acids. Synthesis of backbone of tags and PLs, or triacylglycerols and phospholipids. Here we concentrate on the head groups of tags and PLs. Most glycerol 3-phosphates come from siphoning off DHAP from glycolysis. Remember that glucose, the 6-carbon molecule, generates two 3-carbon molecules, one of which is DHAP. DHAP is reduced by glycerol 3-phosphate dehydrogenase to generate L-glycerol 3-phosphate. L-glycerol 3-phosphate is also made by phosphorylation of glycerol by glycerol kinase in liver and kidney. Synthesis of phosphatidic acid occurs before tags. Phosphatidic acid is the precursor to tags and phospholipids. Acetyl-CoA synthetase attaches a CoA to a fatty acid to generate fatty acyl-CoA. The fatty acid is then transferred to the L-glycerol 3-phosphate. This process is repeated to generate phosphatidic acid. Phosphatidic acid can be made into tags or phospholipids. Phosphatidic acid can be modified to form phospholipids or tags. Phosphatidic acid phosphatase removes the 3-phosphate from the phosphatidic acid to generate 1,2-diacylglycerol. The third carbon is then acylated with a third fatty acid to generate a tag. Alternatively, the phosphate of phosphatidic acid can be replaced with the head group to generate the phospholipids you saw in Biochemistry 1. Regulation of triacylglycerol synthesis by insulin. You should have seen this in Biochemistry 1. Insulin indicates that a high concentration of glucose is present in the blood, and that cells should absorb glucose for glycolysis. Insulin also activates the phosphatase responsible for dephosphorylating and activating ACC. This results in acetyl-CoA synthesis and fatty acid synthesis. Lack of insulin results in increased lipolysis, fatty acid oxidation, acetyl-CoA synthesis, ketone body formation, and failure to synthesize fatty acids. In summary, insulin stimulates conversion of dietary carbohydrates and proteins to fat. Individuals with diabetes mellitus either lack insulin or are insensitive to it. This results in diminished fatty acid synthesis, and the acetyl-CoA arising from catabolism of carbohydrates and proteins is shunted instead to ketone body formation. People in severe ketosis smell of acetone. Triacylglycerol breakdown and resynthesis creates a futile cycle. Lipolysis is the lysis of tags to glycerol and 3 fatty acids. The majority of free fatty acids released by lipolysis are re-esterified to form tags rather than being used for fuel. The triacylglycerol cycle. In mammals, triacylglycerol molecules are broken down and resynthesized in a triacylglycerol cycle during starvation. Some of the fatty acids released by lipolysis of triacylglycerol in adipose tissues pass into the bloodstream, and the remainder are used for resynthesis of triacylglycerol. Some of the fatty acids released into the blood are used for energy, in muscles for example, and some are taken up by the liver and used in triacylglycerol synthesis. The triacylglycerol formed in the liver is transported in the blood back to adipose tissue, where the fatty acid is released by extracellular lipoprotein lipase, taken up by adipocytes, and re-esterified into triacylglycerol. Glyceroneogenesis makes DHAP for glycerol 3-phosphate generation during tag cycle. During lipolysis, which is simulated by glucagon and epinephrine, glycolysis is inhibited in specific tissues. Thus, DHAP is not readily available to make glycerol 3-phosphate. Adipose cells do not have glycerol kinase to make glycerol 3-phosphate on site. They rely on glyceroneogenesis to generate glycerol. This pathway uses some of the steps involved in gluconeogenesis. It converts pyruvate to DHAP. Glyceroneogenesis. This pathway is essentially an abbreviated version of gluconeogenesis, from pyruvate to dihydroxyacetone phosphate, DHAP, followed by conversion of DHAP to glycerol 3-phosphate, which is used for the synthesis of triacylglycerol. Biosynthesis of membrane phospholipids. This process begins with phosphatidic acid or diacylglycerol. The phospholipid head group is attached to a diacylglycerol by a phosphodiester bond, shaded in light red, formed when phosphoric acid condenses with two alcohols, eliminating two molecules of water. Attaching phospholipid head group requires activation by CDP. You should remember seeing chemistry very similar to this in Biochemistry 1. In both of these reactions, CMP acts as the leaving group. CDP plays a role very similar to UDP in glycogen synthesis. It activates the molecule and acts as a good leaving group for a condensation reaction. Phospholipid synthesis in E. coli. There are two pathways that start from the same molecule. A head group, either serine or glycerol 3-phosphate, is attached via CDP diacylglycerol intermediate. For phospholipids other than phosphatidylserine, the head group is further modified. In the enzyme names, PG represents phosphatidylglycerol, PS phosphatidylserine. Synthesis of anionic phospholipids in eukaryotes uses similar strategies to that of E. coli. To generate cardiolipin, phosphatidylglycerol displaces the CMP from CDP diacylglycerol. To generate phosphatidylinositol, inositol displaces the CMP from CDP diacylglycerol. Synthesis and transport of cholesterol, steroids, and isoprenes. These compounds are chemically related and distinct from tags, phospholipids, sphingolipids, and plasmalogens. You will notice the shared chemical relationship when we look at the intermediates of cholesterol synthesis. Overview of eukaryotic cholesterol biosynthesis. The names of enzymes involved in these four steps have been excluded, but we will look at them in more detail later. Step 1. 3 acetates are condensed to form mevalonate. Step 2. Mevalonate is converted to phosphorylated 5-carbon isoprene. Step 3. 6 isoprenes polymerize to form the 30-carbon linear squalene. The isoprene units in squalene are identified by the red dashed lines. Step 4. The squalene cyclizes to form the 4 rings that are modified to produce cholesterol. Step 1. Formation of mevalonate from Acetyl-CoA. 3 Acetyl-CoA are condensed to form HMG-CoA. You've seen HMG-CoA synthesized in Biochemistry 1 when you were discussing ketone bodies. HMG-CoA is reduced to form mevalonate using 2 NADPH molecules. HMG-CoA reductase, the enzyme responsible for this step, is a target for cholesterol-lowering drugs because if you can't make mevalonate, then you can't make cholesterol. Conversion of mevalonate to activated isoprenes. 3 phosphates from 3 ATP molecules are sequentially transferred to mevalonate to generate 3-phospho-5-pyrophosphomevalonate. Decarboxylation and hydrolysis of the phosphate creates a diphosphorylated 5-carbon product named isoprene. This is a high-energy compound. The isoprene exists in equilibrium between the delta-3 isopentylpyrophosphate, or IPP for short, and dimethyl allyl pyrophosphate, or DMAPP for short. Step 3. 6 activated isoprene units condense to form squalene. The two isoprenes join head to tail to displace one set of diphosphates. The 10-carbon molecule is called geranyl pyrophosphate, or GPP for short. GPP joins another IPP to form a 15-carbon molecule called farnesyl pyrophosphate. Two farnesyl pyrophosphates join head to form phosphate-free squalene. The bond formed is in the center of squalene. This step requires NADPH consumption. Step 4. Conversion of squalene to 4-ring steroid nucleus. Squalene monooxygenase adds one oxygen to the end of squalene chain to form squalene 2,3 epoxide. Here, pathways diverge in animal cells versus plant cells. The reason why this is called the steroid nucleus is because it acts as a precursor to steroids. The cyclization product in animals is lanosterol, which converts to cholesterol. In plants, the epoxide cyclizes to other sterols, such as ergosterol. Fate of cholesterol after synthesis. In vertebrates, most cholesterol is synthesized in the liver. Then the cholesterol is exported. Cholesterol is exported as bile acids, biliary cholesterol, or cholesterol esters. Bile is stored in the gallbladder and secreted in the small intestine after a fatty meal. Bile acids such as taurocolic acid emulsify fats. They surround droplets of fat, increasing surface area for attack by lipases. Other tissues convert cholesterol into steroid hormones and so on. Modifications of the cholesterol structure are shown in red. Esterification converts cholesterol to an even more hydrophobic molecule for storage and transport. Each of the other modifications yields a less hydrophobic product. Cholesterol and other lipids are carried on lipoprotein particles. You've seen this before in Biochemistry 1. Lipids are carried through the plasma in spherical particles where the surface of the particles are made of proteins called apolipoprotein and a phospholipid monolayer. This makes the exterior of these particles hydrophilic and their interior hydrophobic. The interior contains cholesterol, tags, and cholesterol esters, which are more nonpolar than cholesterol. Apolipoprotein B100, or ApoB100 for short, is one of the largest single polypeptide chains, containing 4,636 amino acid residues and a molecular weight of 512,000 Daltons. 4 major classes of lipoprotein particles. These particles are named based on the position of sedimentation in isopycnic centrifugation. The composition of particles vary between the 4 classes. The 4 classes are chylomicrons, very low-density lipoprotein, low-density lipoprotein, and high-density lipoprotein. LDL, or low-density lipoprotein, are called bad because they have the highest concentration of cholesterol. Electron microscope pictures of lipoproteins. These are from negative stained grids. Chylomicrons are 50 to 200 nanometers in diameters. VLDL are 28 to 70 nanometers in diameters. HDL are 8 to 11 nanometers, and LDL are 20 to 25 nanometers. The particle sizes given are those measured from these samples, but note that the particle sizes vary considerably in different preparations. Remember, you're separating based on density when you use isopycnic centrifugation, not size, not mass. This next series of slides discuss the structure and function of yeast acetyl-CoA carboxylase, ACC for short. Use these slides as a guide for how I think the manuscripts you've been assigned should be presented. Please note that I'm not requiring you to follow this format. Simply use it as a guide. Also, I will not be narrating over these slides to give you the opportunity to understand and appreciate that the images are supposed to speak for themselves. And you could use a small amount of text to provide some additional guidance for the reader.